So with respect to ONC201, after discovering it through this phenotypic screen, we worked with a number of different academic investigators, many of which are represented in this room, to iron out the mechanism of action of this drug, for all the way from execution of cell death involving apoptosis, upstream to the signaling pathways laid out on this slide that are manipulated by the drug to cause that phenotype. And then finally, at the beginning of last year, we identified the binding target of this molecule as being a G protein coupled receptor, specifically dopamine receptor D2 or DRD2. So with DRD2, all the prior DRD2 antagonists that have been developed as antipsychotics are competitive antagonists. So that means they stick directly in the native ligand binding site, which for this receptor is where dopamine makes contact. That is critical for maximum therapeutic efficacy. You want to have a DRD2 antagonist that can stick in the center of this GPCR that's buried in the membrane and attack the residues that are critical for ligand binding. So what we found is that ONC201, in contrast, involves these orthosteric binding sites. So if I now play the animation, you'll see a number of purple residues being highlighted. These are the residues within DRD2 where if you make a single point mutation there, it ablates the function of ONC201, its ability to antagonize the receptor while leaving the receptor's functionality otherwise intact. And so what we found was that there's a number of these residues that surround the orthosteric binding site, but there's also a couple of distal residues you'll see lying in allosteric pockets. So this was a relatively novel finding for DRD2. As I said, all the previously developed DRD2 antagonists just essentially stick in the blue space that's represented there. And this idea of contacting both orthosteric sites and allosteric sites as an antagonist is collectively referred to as bitopic antagonism.
And so DRD2 is overexpressed, I listed here to make it easier, in glioblastoma, low-grade glioma, colon cancer, rectal adenocarcinoma, and so on and so forth. These are cases of overexpression. Certainly does not mean that in other cases may also be expressed. But that matches very well with the emerging clinical activity of blocking DRD2. Represented in a different way, so this will be, you will see again that this in red will be in the tumors. DRD2, keep in mind this is logarithmic scale, okay? Don't think that these are very similar. This is logarithmic scale. This is really highly expressed in many, many cancers. That is what makes DRD2 as relatively unique candidate in some cancers.
If you simply survey human tissues that express DRD2, it turns out that natural killer cells uniquely in the immune compartment express this receptor, and it's a negative regulator of their function. So NK cell activation for an inhibitor would be posited as being the pharmacodynamic effect. So again, that DRD2 expression exists on natural killer cells had already been established. The issue was in humans receiving therapeutic doses in the phase 1 study, could you demonstrate evidence of NK cell activation or increase in numbers for that matter? And both, in fact, were seen. You see this very consistent increase, about threefold increase in the median number of circulating NK cells. Now, how much of this is margination versus proliferation is, of course, an issue that would require a more complete analysis of tissue distribution of NK cells, most importantly, which ones are going into the tumors. But gratifyingly, there's markers of activation, granzyme most notably on these NK cells. So it would appear, in fact, that this mechanism is being engaged at the doses and exposures that are being explored.